Hi, my name is Mariana Mateos. I'm at the Department of Ecology and Conservation Biology at Texas A&M. Today I wanted to present an update on the status of Poeciliopsis jackschultzi, mostly in requests for help. Poeciliopsis jackschultzi was recently described a year ago from the Rio Concepcion in Sonora, Mexico. And actually, Kevin Conway, the lead author in the description that's shown here, presented this 1 year ago here at TFC. So it's only known from the Rio Concepcion Basin, just south of Nogales. And here's a type locality image. And this is Kevin transferring the type specimen to Hector, so it would be housed at UNAM. So I wanted to highlight that this species has a really small distribution. So I'm just zooming in Google. As you can see, Tucson up here, Hermosillo's down further south, and here's the town of Imuris. So those dots that I'm showing is the approximate known distribution, at least at some point. And if we measure the distance between those in a straight line, it's only 35 kilometers. But it's not found constantly throughout this area. It seems to prefer spring-fed marshy streams and pools with very low depth and very low or no current velocities. This is the bridge just under Imuris, the Highway 15. So we know that at least one of the sites where it was actually most common in the 80s has been obliterated by the expansion of Highway 15. This is based on Bob Reinhoek's recollections. And in terms of threats, of course, this is a desert region. There's limited water, so groundwater extraction and pollution, from human waste or agricultural activities are threats, and invasive species, including the mosquito fish and green sunfish, have been recorded in this area. It was last surveyed in the early 2000s, and it was really hard to find, so it was very uncommon already. Based on the data we have estimated that its extent of occurrence is only about 7 square kilometers, and its area of occupancy is only about 12 square kilometers. So these are measures of the IUCN. So we haven't really done much except try to list it under IUCN, and its publication in the list is pending. The expected category will be endangered, and I want to thank Tim Lyons for facilitating and compiling this, and Dean Hendrickson for serving as a reviewer. I'm just waiting for the Mexican government's call for proposals to modify its Norma Oficial 059, which is the list of endangered and threatened and protected species. I do not know when it will be released, so if anybody here does know and hears, I would really appreciate that you send me an email, because I have no way of, I guess, monitoring it. In the plans, we were going to survey it last May with Doug Duncan, who had agreed to help, but of course our plans were derailed by the pandemic. So we're still hoping that we will be able to go sample in the near future. And again, anyone who wants to join in is very welcome and encouraged. We need to secure funding, of course, and at present my immediate plans is to include aspects of the conservation of the species as broader impacts of an NSF proposal on asexual Poeciliopsis. But again, any other suggestions of sources of funding would be greatly appreciated. Of course, if we find it extant, then we'll need to begin conservation efforts. To be honest, I don't really think I should be leading this, so if anybody wants to take the lead, I will be more than happy to provide a supporting role. It's possible that captive breeding would be beneficial. I mean, they're like, other Poeciliids, and they're not very difficult to captively breed. And in terms of maybe some of the strategies to preserve it once we find it, maybe encourage the establishment of these private reserves, given that it's small range. These known as áreas destinadas voluntariamente a la conservación in Mexico. And of course, we'll be in need of local outreach. So Imuris is the closest, biggest town, but there's several nearby communities. What makes it a little bit more complicated, and in a way, part of the reason that it was difficult to describe, is that morphological identification of specimens is practically impossible. So let me explain why. So here's what we know about the phylogeny, or at least the best phylogenetic hypothesis to date. So this is of the genus Poeciliopsis. Don't worry about the color codings, they're more relevant for biogeography. But Poeciliopsis jackschultzi is up here in the Leptorhaphids group. Its relationships to the other members of Leptorhaphids are not very clear. It's sort of messy in here. And it falls within the, what we know as a predominantly northern clade of Poeciliopsis, so north of the majority being north of the Trans-Mexican volcanic belt. I'm highlighting that it's closely related to the Gila Topminnow, and of course the Sonoran, the Yaqui Topminnow, which I'm not showing here, but sister to the Gila. And I'm highlighting here another relevant species to the story, which is Poeciliopsis monaca, or the headwater Topminnow, which is the mother of all asexual hybrids of Poeciliopsis. So I'm going to focus on just this group in the next slide. So again, the predominantly northern group. So in terms of distribution, so the Gila Topminnow occurs with the Rio Concepcion Topminnow in the same localities. So there's sympatric there. And Poeciliopsis monaca, although it's not sympatric, it actually is found, I guess the closest sexual populations are over 400 kilometers south in the Rio Mayo. But its hybrids are present in these sites. So first, concerning jackschultzi and occidentalis, two sexual species, with the limited markers that we have examined, there's no evidence of hybridization between them, but this really hasn't been fully addressed. So that's a possibility. And of course, we will be using, or planning to use, genomics approaches to check for evidence of this in both directions, towards jackschultzi and towards occidentalis. And concerning the monaca, the headwater Topminnow, or at least its genomes, let me explain. So, monaca, essentially the genus Poeciliopsis is probably most well known for the fact that it has several types of asexual hybrids. These asexual hybrids all ultimately derive from female Poeciliopsis monaca. So here's one example. So in the Concepcion we have monaca occidentalis, So it's a result ultimately of a cross between a female from Poeciliopsis monaca and a male from Poeciliopsis occidentalis. And we also have molecular evidence of monaca jackschultzi hybrids, so which I'm showing here in green, and I'll just refer to them as MJ. Monaca occidentalis are all female. Monaca jackschultzi, I have only found females, but there's been reports of some males in the past, so that awaits to be confirmed. So in terms of the challenges, I guess the logistical challenges to study this species is that at that localities where jackschultzii is present, we have 4 different types of females. So 2 sexuals, jackschultzi and occidentalis, and then 2 asexuals, 2 hybrids, which are going to have intermediate phenotypes, which makes it very challenging. So you essentially at this point must use molecular markers. I think once we have sampled substantially and genotyped substantially, maybe we will find morphological traits that are clearly diagnostic. So hopefully we'll get to that at some point. So I wanted to explain what I mean by asexual and how this works. So the mode of reproduction of Poeciliopsis monaca occidentalis, which has been well studied, is known as hybridogenesis. So the way it would have started is you'd start with a sexual female monaca, which is diploid, and each chromosome set indicated by an M here, and the sexual Poeciliopsis occidentalis male, again diploid, O's representing each of its chromosome sets. This would undergo normal meiosis and form an egg, haploid egg, M, and that female would have mated with occidentalis and generated an F1 hybrid, that has monaca and occidentalis genomes. It's a diploid. What happens is once this hybrid is formed, it goes abnormal meiosis and undergoes abnormal meiosis. And this abnormal meiosis excludes the paternal genome. So basically the O chromosomes are kicked out and not inherited to the next generation. So the M part, the maternal derived and the monaca derived part, is the one that's passed along intact. So that's why I'm showing here this asterisk sort of indicating that these two are genetically identical eggs. The lineage is maintained by repeated, essentially back crossing to occidentalis and so forth. So again, the M is transmitted intact. The O is replaced every generation, and this prime is sort of indicating maybe a slightly different genotype from this one, and it's expressed in the offspring, but then not transmitted. This also indicates then that all asexual females are going to carry mitochondria derived from monaca because mitochondria are maternally transmitted. So here I'm showing cytochrome B haplotype tree, so mitochondrial haplotype tree, of all the monaca and asexual samples to date. So I've sort of divided the haplotype network into 2 regions. This one that's grayed out is basically some samples of Poeciliopsis monaca and asexual biotypes that involve Poeciliopsis lucida, not occidentalis, in the Rio Fuerte and further south. So, and I purposefully made the font small. You don't need to read it. And then on this side in yellow, I'm highlighting those that are essentially from the Rio Mayo, and up north to the Concepcion, all the monaca and asexual biotypes that involve occidentalis, and as I'm showing, also jackschultzi. So in this next slide, I'm going to expand this part and pretty much ignore this one. I'm just going to read the code quickly. So black font means it's pure Monica. So I'm indicating here, for example, or some here. And then blue font refers to Monaca occidentalis, and green font refers to Monaca jackschultzi, and you can ignore the other colors for now. So again, zooming in on this part of the tree, so far we have 4 haplotypes, mitochondrial haplotypes found, and we have this A haplotype here, which is widespread. It's found pretty much in all of the rivers, from the Mayo to the Concepcion, which is the northernmost. And it is found in both in Monaca in the Mayo, it's found in Monaca occidentalis in all of the rivers, and it's found in Monaca jackschultzi and the Concepcion. This one, for example, C, is found only in the Yaqui so far, and this one, B, is found only in the Mayo so far in Monaca occidentalis. And then we have a haplotype that's actually endemic to the Rio Concepcion, and that one is found in both Monaca occidentalis and Monaca jackschultzi. So what this is suggesting is that essentially the Monaca occidentalis and the Monaca jackschultzi share the same Monaca-derived haplotype. So that suggests that this is probably what's happening. So here I'm showing a Monaca occidentalis female, so she's asexual, and as I said before, she would produce pure M haploid eggs. So I'm sort of showing one to the left and one to the right. I'm distinguishing the genome derived from the different species with colors. So black for M for Monaca, blue for O occidentalis, and green for jackschultzi. So in this case, this egg is fertilized by occidentalis sperm. So again, we just recover diploidy, and it's just essentially an F1 hybrid again, Monaca and occidentalis, and again the M part is cloned and so forth. The story goes. On this side, all we've done is replaced occidentalis with jackschultzi, so now we have a jackschultzi sperm, and now you have an MJ. But she'll produce M eggs, and again they could be fertilized by either occidentalis or jackschultzi sperm. So we are not certain that these are hybridogenetic, but the evidence we have to date suggests that they are. As I mentioned earlier, there were some reports of males sometimes, so it's possible that asexuality can break down into basically revert to sexuality, but we don't know. It's something that needs to be investigated. I think the interesting and relevant questions to address of this system, of course, find out whether jackschultzi is extant, and I really hope it is. And at least we've taken the first steps so that it can be protected. Again, without description, it couldn't even be protected. And establish whether it actually occupies, particular microhabitats. It would be important also to determine whether it has an admixed genome. Has it received introgression from occidentalis or vice versa, or even from Poeciliopsis monaca? If asexuals were, to revert to sexual reproduction, that could occur. And from a conservation genetics perspective, we definitely will want to know what are its levels of genetic variation, whether there's any structure among, any localities where we find it. Also, understanding its evolutionary ecology. I think it's, interesting from a basic perspective, but also relevant to conservation. Is there any evidence of niche partitioning among the different, the different asexuals and the two sexuals? I think this raises an interesting question. So, as I said, if these females can obtain sperm from different species, remember that their phenotype is expected to resemble something between, the maternal Monaca and the paternal. So You know, I would probably hypothesize that jackschultzi males probably prefer pure jackschultzi females, and of course that's going to be in their best interest because their sperm is pretty much wasted when it ends up in a hybrid. But then we would expect that Monaca jackschultzi resemble jackschultzi more than Monaca occidentali, so maybe this would be the direction of preference. And similarly, in occidentalis, we may find an opposite pattern. So, of course, the occidentalis females preferred, but then after that, probably the Monaca occidentalis followed by these. I think this raises interesting questions, also relevant for conservation. And finally, just also understanding what interactions the species has with other native and non-native species. So I do want to acknowledge, Bob Ryan-Hoke, my PhD advisor, he discovered jackschultzi, a combination of allozyme work and morphology work, but he thought it involved an asexual that had reverted to sexual reproduction, and all the evidence to date suggests that it's not that. And two of his former students, Ori Sanger and Margaret Skang, Ori did the early DNA work in in her dissertation of 1998, and Margaret did early allozyme work in her master's thesis in 92. Neither of these had been published. Of course, Alejandro Varela has been instrumental, I think, throughout all these years, helping with fieldwork. Also, Dean Hendrickson helping with fieldwork. And actually, I thank all three of them, including Gary Meffey, who were very gracious for me to pick their brain on details about localities from way back. Of course, I really want to thank Kevin for enabling essentially the next steps with the species, basically doing the morphological description, and funding from acknowledged funding from NSF way back, as well as from the from Mbari while I was a student at Bob's lab. So hopefully this will help recruit and find collaborators who might contribute to preserving the species. So I want to end with a plea for help from the Guatopote del Concepcion. It would be, I think, a shame if it went extinct, being that is found in a pretty accessible area, so again, not too far from the main federal highway. If you have any information or need any information, my contact information is here, and I want to thank you for your attention.